The overall goal of this protocol is to enable clinical support staff in a dermatology practice to effectively process and store cutaneous tumor samples for downstream laboratory applications without interfering with routine clinical operations.
This protocol was designed to enable medical assistants and Mohs histotechnicians to collect and store cutaneous tumor samples for future biomedical research projects as part of their daily clinical operations.
The main advantage of this protocol is that excised tissues that are normally discarded and destroyed are instead collected and preserved for research using procedures which minimally impact routine clinical operations.
The variety of samples collected enables correlations between protein and RNA biomarkers in the tissue and plasma samples with patient outcome data.
In principle, this protocol, when slightly modified, is applicable to any surgical practice that routinely discards patient samples gathered in the course of patient treatment.
Demonstrating the procedure will be Stephanie Fox, a Mohs histotechnician, and Dr. Mitchell Manway, a research fellow in our practice.
To begin, use the tissue received from the Mohs surgeon and with a scalpel, excise a 10 to 50 milligram sample from the center of the apical side of the tissue. With tweezers, transfer the viable tissue to a labeled 1.5 milliliter tube pre-filled with culture medium. Make sure the tissue is entirely immersed in the medium. Place the sample in a refrigerator and store it at 4 degrees Celsius for no longer than 24 hours before further processing.
Harvest at least a 10 cubic